The presentation on the origin and continuing evolution of SARS-CoV-2, written by Zhaolu Tang, Cheng Wu, Zhang Li, and et al., presented by Eli Fox and Zane Van Meter, under the supervision of Emily Berglund. Alright, so a little background for you guys on the current status of the SARS-CoV-2 virus, if you guys aren't following religiously. There are currently just over 3 million confirmed cases throughout the world, with a third of these cases from the United States alone. Additionally, there are 216,160 confirmed deaths. And just to end on a positive note, there are 921,199 recoveries thus far. So while SARS-CoV-2 is the latest update in coronaviruses, several teams have recently confirmed genetic similarity between other coronaviruses, as well as suggested that it was derived from a bat reservoir. Using whole genome sequencing, the novel virus has 96.2% similarity to bat SARS-related coronavirus, denoted by RATG13. Its next most related coronavirus is SARS-CoV, which has about 79% similarity and has been confirmed to use the same receptor, angiotensin converting enzyme 2, to mediate cell entry. Additionally, MERS-CoV is the third most related coronavirus to the novel one, with 50% genomic similarity. So throughout this paper, there's some terminology that you'll need to be familiar with if you are not already. The first being open reading frames, or ORFs, which are the regions of RNA in this case that can be translated. The article also refers to non-synonymous substitutions and synonymous substitutions. So non-synonymous substitutions being a single nucleotide substitution or deletion that causes frame shifts, ultimately altering the reading frame. And synonymous substitutions, on the other hand, are just changes in one base pair rather than an insertion or deletion. SNPs, or single nucleotide polymorphisms, is a substitution of a single nucleotide that occurs at a specific position in a genome. And finally, dN over ds, also denoted by omega, which is used to estimate the balance between neutral mutations, negative selection, and positive selection within protein encoding genes.
Alright, so now we're going to cover the methods section of the study. So first up is data extraction. SARS-CoV-2 data was gathered from a database called the Global Initiative on Sharing All Influenza Data, or GIS-AID for short. I'm just going to call it that from here on. As an interesting anecdote, this is actually the same source that sites like nextstrain.org use. I just thought that was interesting. From this database, 103 complete SARS-CoV genomes were obtained. Comparative genomes of other viruses were obtained from either GIS-AID or GenBank. Researchers also used annotated coding domains of other viruses. And so to ensure that none of these were missed, researchers went back through and observed open reading frames of viruses, and they were annotated using coding regions of SARS-CoV. So for data analysis, several programs were used in sequence alignment. These include programs by the name of Muscle, Exonerate, and RevTrans. So essentially, these serve a similar function to that of an NCBI BLAST, which, as we are all familiar with, allows you to compare multiple sequences. But of course, as you might imagine, they allow for more in-depth analysis in sequence alignment, and beyond that, they allow you to do further analyses. Creation of the phylogenetic tree was conducted in a program called MEGA-X. Beyond this, several other analyses were conducted. They looked at positive selection of amino acids, haplotype alignment. This was done to construct the most likely phylogenetic tree of SARS-CoV-2. SMP calling, and finally, codon usage bias analysis. So of course, each of these analyses included a variety of programs for each. We're just not going to go into depth on that.
So, the importance of the trimeric spike protein cannot be overstated as it mediates cell-to-cell contact, initiating membrane fusion and entrance to the human cell via ACE2 of the host cell. Analysis of the amino acid residues in this research paper give insight into its somewhat muddled evolutionary history, finding that the 6 critical amino acid residues present in the receptor binding domain in spike of SARS-CoV-2 are also found in a coronavirus present in pangolins. And so that is a pangolin in the bottom right, and I think they're kind of cool because they look like claymation. But anyway, this similarity was first thought to be a result of a recombination event. However, after analysis to calculate divergence time, which is denoted by t in the equation below, it is thought that if the recombination event did occur, it would have happened approximately 19.8 to 55.4 years ago. And due to how recent this recombination event would have occurred, it suggests that the identical function of amino acid residues between these two viruses is more likely a result of coincidental convergent evolution rather than a recombination event. Alright, so this article also kind of analyzed the selective constraints of the evolution of SARS-CoV-2. And to determine these selective constraints, a couple of methods were used. Strong indications of negative selection was found for non-synonymous mutations during viral evolution, as the genome-wide omega value between SARS-CoV-2 and the other coronaviruses ranged from 0.044 to 0.124, which you can see in the bottom of the table in the average column. And this can be interpreted as an 87.6% to 95.6% removal of non-synonymous mutations by negative selection. In determining the prevalence of positive selection during the viral evolution of SARS-CoV-2, coding sequences of the 9 conserved open reading frames from each virus were compared using an M8 model and code ML. This comparison yielded 1.52% of non-synonymous traits being positively selected, with the remaining 98.48% occurring under neutral evolution or negative selection. So while these coronaviruses were generally under fairly strong negative selection, analysis of 10 amino acid sites showed strong signals of positive selection. Interestingly, these 3 sites of positive selection were located in the receptor binding domain of spike, as you can see below. So, an attempt to determine if recombination occurred among 103 SARS-CoV-2 virus strains, HaploView was used to analyze patterns of linkage disequilibrium between alleles. And while no recombination was observed, SNPs at 2 locations appear to have a very strong linkage. The graph on the right side of the page depicts this strong linkage with an R-squared of 0.954. And of these 103 strains of SARS-CoV-2, 101 of them actually had complete linkage between the 2 alleles. 72 of the 100 strains sampled were designated as L-type, as the linkage at the nucleotide 28,144 was in the codon of leucine, and 29 of the 101 strains, however, were designated as the S-type because the linkage at the nucleotide 28,144 occurred in the codon of serine.
Okay, so now we'll get into the discussion of the study. So while SARS-CoV-2 was found to be closest in relation to that of the RATG13 virus, This difference is underestimated when synonymous and non-synonymous sites are not separated. So this is because non-synonymous sites undergo more negative selection than synonymous. So basically, to demonstrate this underestimate in sequence difference, we can consider these two closest sequences, that of SARS-CoV-2 and RATG13. So we'll consider these two viruses. The non-synonymous value between the two, essentially the difference value between the two, is 0.17. Now, for reference, the non-synonymous value between the complete human genome and the chimpanzee's complete genome is only 0.012. So this means that the molecular divergence between the two viruses is actually 14 times greater than that of human and chimpanzees. So another finding was that selection in SARS-CoV-2 seems to be undergoing more negative selection than positive. An additional finding is the identification of two separate strains of SARS-CoV-2. These being the L and S strain, as we covered. The S strain is the ancestral strain of the two and is named for the mutation on the open reading frame 8 that results in a serine. The L strain is derived from the S and is named from the mutation in the open reading frame 1AB and results in a leucine. It's unclear if the occurrence of the L strain arose from within humans or it occurred actually in an intermediate host. The L strain also seems to be more aggressive and seems to have increased in relative abundance around the time of the outbreak. So moving on to conclusions. So overall, the largest takeaways from this study have to be finding the L and S strains of SARS-CoV-2 and the discovery of the bat virus RATG13, and that basically it's the most similar to that of SARS-CoV-2. However, as we just stated, this finding has large limitations to that. Thanks to the study, there's really primarily one prominent strength, and that has to be the identification of the L and S strains and the methods that were used to come to the conclusion that of their existence. So the first analysis used to identify the L and S strains was the SNP analysis, and this labeled the two novel groups by display of their two distinct mutations. Of course, the L group being the leucine and the S group being the serine. This finding was also corroborated by their other analysis, the haplotype network using these SNPs. So ultimately, the use of two separate tests which both reach the same conclusion certainly lends more validity to the finding. Now we're going to move on to the study weaknesses. So drawbacks of this study are, of course, the fact that it has become quickly outdated. As of today, nearly 2 months after the study, there has been the identification of many new mutations, both in nucleotides and amino acids. Another weakness is, as stated in the paper, the sample sizes that analyses were conducted was very limited. So the data that they had at their disposal was not very large, and the takeaways from that and the conclusions basically reached by the paper should probably be retested over larger sample sizes and data sets just to ensure their validity and the same conclusions are reached over larger, more powerful data sets. The final one is maybe not necessarily a weakness, but that this paper is certainly written for an audience familiar with the many analysis methods and software used. So that being said, we're not necessarily certain on the validity of the methods used, just meaning we're not sure whether or not they used the correct um, kinds of analyses and tests, so we are unable to um, speak on their choice in analysis. So now we can look at study implications, or basically the impact of the findings within this study. So the presence of novel strains of coronavirus suggests that there may be discrepancies in disease presentation from the virus, being that the L strain seems to be more virulent than that of the S. This also suggests that there may be regional variation in the severity of disease due to which strain of the virus is more abundant in a given area. So that particular finding would obviously impact anybody within an area that an L strain is in more abundance, especially physicians and policymakers in that given area. In addition, the evolutionary similarity in that of SARS-CoV-2 and the bat virus RATG13 reaffirms the the possible source being that of bat or bat origin. This finding certainly helps in identifying the organism involved in spillover, and certainly could aid in motivating future efforts in limiting spillover events, like that of SARS-CoV-2. So finally, we're going to look at some questions that we have following the study. Our most immediate and was recognized by those who were writing the study was that the study repeated with more current data, or at least larger data sets, might yield other results. And after reading the study, that was kind of our primary question, whether or not the same conclusions would be reached with larger data sets, that being the finding of L and S strains, as well as with current data maybe identifying the possibility of even more strains having existed. Another question we arrived frequently on throughout the article is, how do these methods differ from how nextstrain.org performs their analysis? So are their methods more thorough? Are they less thorough? Are they maybe the exact same kinds of methods and data analysis? Being that, I mean, they even use the exact same data sets. It would be interesting to know whether or not they differed at all, and maybe even how they differed. Kind of on this point, GIS Aid themselves released a statement cautioning the use of their SARS-CoV-2 data in the identification of different, what they called either types or strains of SARS-CoV-2. They say that this is because these differences that are observed in the short term may be actually just due to founder effect and not evolutionary pressures at large. So again, it would be interesting to know if this study is comprehensive enough to not be limited by the founder effect, and if the strains identified in nextstrain.org are as maybe genuine as the strains identified in this study, and again, if the strains identified in this study are even valid due to that of the founder's effect.